And our first speaker is going to be Travis Lee, who's going to talk to us about spatial omics in plants, the how, when, where, and why. Travis received his PhD at UC Riverside, where he worked with Julia Bailey-Serres. He then subsequently joined Joe Ecker's lab at the Salk Institute in July of 2018, where he's been doing some really amazing work in single cell and spatial omics, looking at seed-to-seed development in Arabidopsis, as well as things like ethylene response. So we're excited for your talk today, Travis. Thanks for joining us. Go ahead and take it away.
Okay, thanks for the very nice introduction, Trevor. So let me see if I can share my screen. I think is that showing the slides? Or is that the
That looks
okay.
like presenter view.
Okay, let me try again. Okay, how
Better,
about now?
better.
Okay, great. So again, thanks for the introduction. So today this will be a little bit more of a practical-based presentation, but what I'll be describing is kind of how, when, where, and why you might want to use spatial omics in any of the plant species. So just beginning with the why, just very quickly, in the recent couple of years or past decade, there's been an explosion in kind of technological advances in the genomics world. So really what we're starting to access is looking at biology in high resolution. So I really like this comparison where when we take bulk sequencing, basically we're just grinding up all of our cells and cell types into a slurry or a smoothie, and we could get a lot of readouts and learn many interesting things. But really, this is a combination of many different cell types. But with the advancement of single cell sequencing, what we can then do is sequence the transcriptome or some sort of genomic output of individual cells and then group them together. So we could say, this group of cells that are orange might be one cell type, whereas another cell type might be yellow. And that's just based on, molecular signatures or transcriptional signatures. But really, as we're moving forward with technological advances, what we're seeing is that with spatial omics, the kind of benefit is that we can now observe, where an RNA is expressed in the native arrangement of a cell within the tissue. And I think that brings a lot of power to plant biology as well. So first, I just like to begin with, there's a lot of different methods available, and this is not a new problem, but really when we're speaking about spatial omics, there are kind of two flavors where we are looking at kind of two dimensions, which is maybe one individual cell layer of a plant or whole mounts. And this goes back to, in situ, original in situ experiments. But really, I think this comes down to what you might want to be looking at as your biological question. Do you need three-dimensional space for your information? Or is it fine looking at just two dimensions? And today I'll be focusing just on techniques that utilize, these thin sections or two dimensions of your plant development. But there are other methods available that does enable, this multiplexed imaging or analysis of individual transcripts, such as a phytomap, among other whole mount methods that are coming out on bioRxiv in publication. But when comparing these two, kind of flavors from 2D to 3D, another question that you might need to ask based on your experiment, what kind of throughput do you need to visualize, your expected output? Are you fine with looking at, about 50 genes or fewer that can have the advantage of 3D? Do you need a higher throughput? Do you need to visualize, hundreds to 1,000 genes simultaneously? And that kind of throughput is where we might, move towards some of these technologies that utilize these thin sections. Additionally, there's inherent properties about whatever sample you may be looking at. What is the size of your sample? What is the shape? How permeable are the cells? Whereby some of these enzymes need to enter the cells or reagents need to enter the cells, and if you can't permeabilize the cells easily, it may not be easily achieved in that assay. So really, when we're talking about these thin sections, a 10 micron is what most of these technologies utilize. Really, we're just taking a very thin slice of our plant tissue. Um, and just the way that you can kind of think of this, we need to start thinking about how this might affect, or what we want to look at individually in our experiments. So really, with these very thin sections, what we should get is, probably one individual cell type, because cells can be, quite much larger than 10 microns. And just using the leaf as an example, if we were to take a 10 micron section of a leaf, and we were to look inside, what we'd see is that, there's a variety of different cell types and cell layers. We have the epidermis, the mesophyll cells, and then, the epidermis, lower epidermis. Um, and if we were to take a cross section like this, we'll get good cell type representation. We'll get all these different cell types. But what we really lose is the positional information. If we take this thin section in the middle of the leaf, what we're then losing is the positional information within the leaf, by not taking a whole mount sample. But if we're really just interested in one individual cell type, such as the palisade parenchyma cells, um, we can also take a longitudinal section, along the leaf, and then we can still capture this kind of gradient of development that is observed in leaf emergence. And what that might look like is just a thin section here, where you'll see basically when you cut this thin section, you'll see the same cell types. In the middle, you'll probably see the vasculature or the veins of the plant as well. But really what we're assaying is just one individual cell type at this, perspective. And this might be important if we were, really investigating something that's developmentally regulated, where we might see differences along the leaf, where that positional information is very important. Or even taking this to a different, type of sample. If we were looking at roots, roots are associated with different zones of development and developmental stages. And again, this might be, one particular aspect where we might want to perform a longitudinal section, and look at, maybe one or two cell types along the gradient of development. So again, this kind of comes back to, what is the biological question and what are the properties of your sample? So just as the general workflow, the how to, like I mentioned, what you need to perform for most of these assays that have been described earlier in the morning very nicely by our speakers, is these thin sections, and that's a bit of a process, but really what that entails is just taking your sample, rapidly freezing it in some sort of embedding medium, and this is to preserve the integrity of the RNA that you'll be looking at downstream. And then cutting that section into a thin section in a cryostat, which is essentially just a temperature-controlled microtome. Um, you know, Once you're in the microtome, you're going to enter your sample and cut a 10 micron thick section that looks like this. And hopefully at the end of this, what you should observe is, for your sample, you should see, intact morphology of your sample. And, just zooming in, you can see that these individual cells seem to be fully intact. And this would be successful. So really, the important part is just getting your sample frozen to maintain the RNA integrity that you'll be looking at downstream, and making sure that the morphology of their cells is fully intact, so that it's not going to be kind of mixing together. Um, and just kind of talking briefly about the technologies, these are some of the methods that we use in the Ecker lab, but there are also other alternatives. But really, this can be split into kind of two categories. If anyone saw the talk earlier, there's the unbiased approach, where this is going to be sequencing based, and what we used was a reagent called the Seeker tile from Curio. And essentially what this entails is very small, a glass slide that has this array of oligos that's attached to it. And basically all these beads, they're very arranged in a very specific manner and very close together with 10 micron resolution. And what this is going to have associated with it is a basically PCR handle and a bead barcode. So we can then look at where the bead is located on that particular slide. And the poly A tail will hybridize to the poly DT, and that's how we can perform our sequencing and determine where that transfer came from. The opposite approach is taking a targeted approach with an imaging-based platform, such as the VIGID Immerscope, but alternatives also exist, such as the 10x Xenium. And this is an imaging-based approach where over several rounds of imaging, oligos are going to hybridize to your RNA of interest, and basically there's going to be a binary on-off, fluorescence. And when you kind of look at that downstream after several rounds of imaging, you'll be able to decode what transcript is where based on this code book. And there's both some advantages and disadvantages to each of these platforms. So this is also something to consider. Where with the unbiased platform, there's no really specialized equipment that's required. You just need to cut your sample and place it onto this slide. You don't need to know what genes you're looking for, and it's species agnostic because it's just targeting the poly mRNA of your sample. But there are also some cautions when considering this, which is, there are some size restrictions. At the time when we performed these experiments, there was only a 3 mm squared tile that was available, which is quite small, and you can't fit a lot of plant tissue on it. But since then, they've released a larger 10 mm squared section or slide. And because this is sequencing based, it may be difficult to kind of capture low expression transcripts because essentially all you're doing is allowing the RNAs of the cells to attach to the tile, and the higher abundance transcripts will, most likely be sequenced downstream. So it may be hard to visualize, maybe your favorite TF that's at a low expression level. Oppositely, with an imaging-based platform, this does have several advantages as well. Such as the detection of low expression transcripts, so we can target individual transcripts, and it's also high resolution. So the output is going to be these spots, which is going to be indicative of an individual mRNA molecule. But there are also several cautions. We must know what targets we want to look at. So having single cell data that has already been generated would be very beneficial. There is also specialized equipment, and downstream, one challenge can be cell segmentation, because ultimately what we want to get at is a cell by gene matrix that's utilized for many of these single cell analyses. And cell segmentation can be a challenge for plants among other species as well. So I'll just be detailing a couple of ways that we've used this in the Ecker lab, both the sequencing-based and the imaging-based platforms. And so we've used this in a project that we've performed, which is generating this atlas of seed-to-seed development at the single nucleus level. So we performed single nucleus RNA-seq of 10 different developmental stages, and this really helped in the annotation and validation of some of our clusters that we observed. Also, we've performed, this assay that's kind of followed pathogen attack in leaves that the next speaker, Alex, will be speaking about. But really what you can also observe is kind of, this heterologous or heterogeneous responses of leaves to pathogen infection that are both available in bioRxiv right now. So just very briefly about the experiment that we performed in the lab for the atlas is we took these 10 developmental stages of Arabidopsis. We performed nuclei extraction and generated single cell sequencing data sets. And how that looked in total was these 10 total data sets with over 400,000 nuclei. And what we found was that some of these data sets, particularly the seed and Silique data sets, these had quite a bit of challenges with the annotation downstream. So we can see that there's many clusters, but saying what that cluster might correspond to was a challenge because in many cases, at the time when we were doing these experiments, there was not a reference single cell data set or not a cell type specific reference data set as well for these particular stages of development that we assayed. So that's kind of the last thing I'd just like to mention is that we also have this all of these data sets available online to browse. at our Arabidopsis Developmental Atlas website. So you can just click on one of these data sets, load it up, and see where your favorite gene is expressed in any of these 10 data sets. So the way that we really utilized these technologies was helping to validate these complex tissues, which is again the germinating seeds and Siliques. So again, several of these lacked reference data sets at the single cell or cell type specific level. Additionally, these organs comprise multiple different tissues as well. So for example, with the seed, what is inside is we have the seed proper and also the embryo within. So that's kind of two different tissues. And also with the Silique, we have the Silique tissue proper, which is the fruit of the plant, and also the developing embryo and seed within. So the complexity of these data sets really kind of annotating the clusters was kind of challenging. So we use both the sequencing and the imaging-based approach. For the germinating seeds, we use the sequencing-based or unbiased approach. Really because the size of the seeds were kind of allowed for this. Seeds are very small, so we could, in this particular example, we kind of made an array of seeds in one of our tissue blocks, and we simultaneously profiled several seeds in one experiment. And conversely, with the mature Siliques, what we chose to do was use the targeted approach because we did see this high level of complexity in the clustering. So we could select many different targets that we were interested in and determine where they're expressed and see what that cluster is associated with. So just how this data looks with the germinating seed, what we can do is, again, we're going to have an output of gene expression. We can cluster the data based on each of these individual spots. So looking at it in the UMAP space, we can see there's these large distinct populations, and when you plot those cluster labels to the spatial information, what we can then start to see is the outline of what we expect to see is maybe the developing seed and also the seed. So these are just kind of zoomed in here of the developing seed. And when we take these different cluster colors, we can see that essentially they correspond to different regions of the developing seedling. So we can annotate, the cotyledons and root tip and maybe the epidermis and the vasculature or stele of the plant. And that's just based on, how the clustering looks and transferring the colors. So at the RNA level, we can again take individual genes such as GSTF9 that's depicted here. Again, showing the same cluster or colors for the clusters between the spatial and the UMAP dimensionality reduction. And we see that this particular gene is expressed down here in what appears to be the seal cluster. And it is enriched in specific regions of our droplet-based data set. So we can, kind of somewhat confirm with high confidence that that region is associated with the stele of the developing seedling. So that was really great and allowed for higher confidence in annotating our seed data set, which maybe lacked some high resolution data sets to kind of correspond to it. And on the other end, performing the spatial investigation of Siliques, what we have is this high level of complexity where we observe 26 different clusters over 70,000 nuclei. And I believe this stems from the fact that we have these different axes of development that are kind of maybe causing these cells to be slightly different from each other. So for the harvesting of Siliques, we actually harvest from younger to older Siliques. So there's this developmental axis of true development. But also there's an elongation axis where the cells at the tip of the base of the Silique may be slightly different from the same cell type in the middle of the plants or in the middle of the fruits. And additionally, just taking a look inside of a Silique, we also have the developing seeds and the developing embryo. So again, we use this imaging-based approach, and what we designed was a 140 gene panel where we can include major cluster markers that looks like this, and as an example, where the expression of this gene is enriched in one of our clusters here, and within our total data set, it's unique to the Silique. And we also took subcluster markers where we can take one of these individual clusters, such as cluster 0. We can take these nuclei and recluster them, and we can still see quite a bit of heterogeneity in this individual cluster. So this is about 10,000 nuclei. And we can take these subcluster markers that correspond to these kind of different regions of the subclustering, where there's some examples down here, where it's localized to this small region on the left, the bottom right, and the top. So we see quite a bit of specificity, and we want to see where these genes might lie, in the actual tissue. So this is how the data looks. So hopefully, we can see what's colored here is about 40 genes of the 140 genes total in the panel. And hopefully what we can appreciate is that these different colors that correspond to, several groups of genes kind of correspond to different regions or cell types or layers of the Silique. And just taking kind of two areas and zooming in on it, we can see that in this yellow area or left area, we can see kind of this layer specific expression pattern of these different colors and that corresponds to different parts of the plant. And also on the right, we can see kind of a zoom in of kind of the structures that we might expect to see within a Silique if you were to cut it open. So just as an example, at the single molecule or single gene level, this is an aggregate of 40 genes. But taking these markers that we observed in the droplet-based data set, what we can see is that we annotated this pink cluster as the embryo, and the colors are swapped, so I apologize for that. But looking at this individual gene within the MERFISH data set, we can see that these yellow dots, which correspond to that same gene, where each individual dot is one mRNA molecule, it's restricted to just the embryo part based on what we see in the DAPI staining. And oppositely, in this other cluster that we've annotated as endosperm, again, looking at where it's expressed, these pink dots, correspond to the gelosal endosperm. So that again helped with the annotation of these data sets. But also, taking these this subclustering, what we really could further look at biologically was kind of where these very similar cell types at the global level of the Silique, what they might correspond to. And what we found was, again, with this subclustering of cluster 0, they actually corresponded to different layers within the seed sac, where we can see that, we've been able to annotate these subclusters as the outer integument layers, the inner integument layers, and flavanol producing cells. And that's just colored here on the individually for all of these different colors and with a composite image. So we can see hopefully that these different colors correspond to different regions and layers of the developing seed. And that's just annotated here. And I think I'm running out of time. So really just some final perspectives about, how and why you might want to perform these experiments. For our purposes, we used it to validate our single nucleus RNA-seq annotations and to potentially get some deeper biological insight in the complex Silique tissues that we saw. But really, I think plant samples do present some unique challenges not present in animal systems, such as the cell wall, but also provide some unique biological insights where the cells do not renew, unlike animal cells. So we actually have this natural developmental gradient that's ingrained in each of our tissues. And really, each tissue may have some different challenges that may be unique. So it's really just kind of perseverance. But really, having some creativity with your sample preparation can also overcome some of these technical bottlenecks that might be present in some of these assays, such as, if it's necessary to, look at both 2D, cross-section longitudinal sections, perhaps they can be introduced together in the same tissue block, or, just kind of, I've used 3D printing as well to kind of help with the downstream application in kind of the arrangement of these samples. So just being creative might help you get to the finish line as well. So lastly, I'd just like to acknowledge my mentor, Joe Ecker, for really, supporting this project and encouraging us to generate these large data sets. And also Tatsia and Nella that were instrumental for the generation of and analysis of the single nucleus data set that I presented, and the rest of the Ecker Lab as well, and HHMR for funding. And thank you.
Thanks, Travis, for this really great talk, highlighting both the techniques as well as the kind of biology we could learn. So I want to remind everybody that you can put your questions in the Q&A box below if you would like to ask them. We do have a few minutes. There's one question already, Travis. So the question asks, could you please elaborate why the tips of the valves are different from the middle? Is that referring to the transcriptomic landscape or something else?
Yeah, so with the Silique development, what actually happens is elongation occurs kind of in the central region of the Silique. So there's kind of different rates of elongation. And also downstream, there's going to be differences at the transcriptional level as well. So that's kind of, I think, where we see all this heterogeneity in maybe even the same cell type in the Silique samples.
Thanks for that, Travis. I was also curious about this 3D printing that you're doing. Can you talk a little bit more about that and how that's been helpful for your sample arrangement?
Yeah, so that's it's just essentially, if you really need to have, your samples arranged very specifically, I think that's what the 3D printing has really just kind of enabled, this very fast prototyping where you can, have a prototype of how to arrange your samples and maybe facilitate kind of maybe different ways to arrange your samples in a array-based kind of platform, where downstream you can section your samples independently. So really, it's just kind of a rapid way to kind of, help with the sectioning, but just taking kind of common lab-based reagents as well. You can kind of just be creative too.
Thanks. It looks like one more came in. It says, if I'm right, do we have to cut the tissue sections of 3mm by 3mm, or I think it's 10mm by 10mm to fit the slide tile? So how does the RNA come out of the cells to bind the oligo-DTB?
Yeah, so that is the 3mm squared is just the size of the slide that is provided by the company, either 3mm squared or 10mm squared. So that's just kind of the total area that you can place onto the slide for your experiments. For both applications, the thickness of the section will be about 10 microns though, which seems to be the standard for many of these, spatial platforms. And the way that the kind of RNA flows onto the slide is, after you place the tissue directly onto the slide, of course, you're going to have that direct application of mRNA to the slide. But there's also steps for proteinase digestion, and also kind of permeabilization that will allow for the mRNAs to flow onto the slide where it can be captured.
Perfect. Thanks, Travis. Well, I suspect we'll be able to continue this discussion later on in the community session. So I think there's also a bit of discussion about the elongation in the chat if you want to take a look at that, Travis. Thank you.
Okay, great. Thank you.